Just sharpen my pencil with a new sharpener. We'll see if it holds up or if it breaks. This is a third pencil sharpener attempt this week. So we're still in glycolysis, and now we're doing the regulating enzymes of glycolysis. Hexokinase converts glucose to G6-phosphate. It's in most tissue. It uses MgATP to native. I don't know how to say that. I mean, pretty sure that's magnesium ATP, but I don't know why they're... Yeah, I need to look into that. It's been a while since I have looked at that chemistry. Okay, so... Put that. Here we go. Hexokinase converts glucose into G6-phosphate, found in most tissue, uses this magnesium ATP sort of situation. It's allosterically regulated enzyme, which is inhibited by its product, which is G6-phosphate. It prevents too much glucose 6-phosphate from accumulating. Too much G6P will cause the cell to be swollen and rupture. Glucokinase does the same as above, as it is an isoenzyme of hexokinase. It's in the liver. It's activated by insulin and inhibits fructose 6-phosphate. It works at relatively high levels of glucose only. It's regulated differently than hexokinase. PFK, or phosphofructokinase, is the most important control point rate limiting step in glycolysis. PFK is inhibited by ATP citrate, which is an intermediate of Krebs, NADH, and by glucagon. This enzyme is stimulated by AMP, ADP, and insulin, phosphofructokinase. So PFK is stimulated by AMP, ADP, and insulin. The reason for regulation at the third step is because G6P is the product of the first reaction, can branch off to other metabolic pathways besides glycolysis. The third reaction, however, is the first reaction is unique to glycolysis and commits the cell to the glycolysis pathway. Pyruvate kinase is the final regulating enzyme activated by fructose 1,6-bisphosphate and AMP. It's inhibited by ATP, fatty acids, alanine, and acetyl-CoA, a major intermediate for catabolism. The fate of pyruvate. Pyruvate is not the endpoint of glycolysis. The cell has a limited supply of NAD+, and without the regeneration of NADH back to NAD+, eventually all NAD+ will end up as NADH, and glycolysis will stop due to lack of NAD+. 4 possible fates of pyruvate. Oxidation, reduction, carboxylation, and transamination. Oxidation in aerobic reactions. Pyruvate is oxidized by acetyl-CoA, which enters Krebs. This is facilitated by pyruvate dehydrogenase, which is inhibited by ATP, acetyl-CoA, and NADH. Thymine is needed for this reaction. Reduction. Anaerobic reactions, a different approach to regenerating NAD+. One pathway converts pyruvate to lactate in a step which produces NAD+. This is facilitated by lactate dehydrogenase. Carboxylation. Pyruvate is converted into oxaloacetate by pyruvate carboxylase. Transamination. Pyruvate may be converted to alanine by alanine transaminase. Glycolysis is not energy efficient. It's not a very energy efficient way to produce energy. It only produces a net surplus of 2 moles of ATP. It produces a total of 4 but consumes 2 in the process, hence the net surplus of 2 ATP. Pyruvate enters the mitochondrion and is converted to acetyl-CoA, which enters Krebs, where a lot more energy can be efficiently produced. Glucose plus 2 NAD plus phosphate plus 2 ATP equals 2 pyruvates plus 4 ATP plus 2 NAD plus 4 ATP plus H2. Let's look through this chart. ATP is used at step 1 along with glucokinase and hexokinase. Phosphoglucose isomerase is used at step 2. Phosphofructokinase is used at step 3 with an ATP. Aldolase is used at step 4. This part of the chart confuses me a little bit, except that I know that DAP needs to be converted. So that's step 5 where we make the DAP. And then step 6, G3P dehydrogenase uses an NADH. Phosphoglycerate kinase uses an ATP at step 7. Step 8, phosphoglycerate mutase. Step 9 uses enolase. Step 10 uses pyruvate kinase and ATP. That's glycolysis. Sorbitol. An alternative method of dealing with glucose in the cell is to convert it to its alcohol counterpart called sorbitol via the enzyme aldose reductase. Some tissues contain sorbitol dehydrogenase, which converts sorbitol to fructose, which can enter the glycolytic pathway by being converted to fructose 1-phosphate and then to glyceraldehyde. Tissues lacking this enzyme are at risk of sorbitol accumulation within the tissue. For example, a tissue that lacks sorbitol dehydrogenase are the lens, retina, and Schwann cells. Interesting. Sorbitol is osmotically active and draws water into the cell. This damages the lens, retina, and peripheral nerves, resulting in premature cataracts, retinopathy, and peripheral neuropathy. Alright, we're not going to start a whole new section, so that's a shorter video.